All right, well, welcome everybody to Grand Rounds. Again, a very special presentation today by Dr. Kelsey Holbert, who is one of our outgoing chief residents and who's going into pulmonary and critical care medicine. Very excited. Just as a reminder, next week's Grand Rounds is virtual, and then in 2 weeks, we will have the last two of our our Grand Rounds, Chief Resident Grand Rounds speakers in person again. So please come and support our outstanding chiefs. And to introduce Dr. Kelsey Holbert will be Dr. Andy Coyle, our Residency Program Director.
Thanks, Lynn. So really excited to announce today's Grand Round speaker, Dr. Kelsey Holbert. So Kelsey is a graduate at the University of Wisconsin-Milwaukee for her undergrad, followed by medical school at the University of Illinois, Chicago. So as you know, she was a resident here at UW. So in addition to excelling in her clinical training during residency, she was active in numerous scholarly projects, one of which we'll hear about today. In addition to being an active participant and leader in our health equity and TEACH pathways. In addition to her research work in pulmonary fibrosis, which we'll talk about today, Kelsey's had a really sharp focus on principles of equity, inclusion, and advocacy. She was a recipient of a medical education innovation grant to build and implement a trauma-informed care curriculum for the residency program, which was incredibly well received, and was a recipient of the 2023 Residency Advocacy Award. As a chief resident, Kelsey has excelled as a teacher, mentor, ally, educator. She has overseen our enormous curriculum, helping to conceptualize, build, organize hundreds of hours of curriculum, all with a really sharp focus on protecting and enhancing the resident learning experience. I can certainly say from our program leadership team, we're all incredibly grateful to Kelsey, to all that she has given to our residency and our community these last 4 years, and is certainly someone we are proud to call a graduate of our residency program. Kelsey will be continuing her training as a T32 research and clinical fellow in pulmonary and critical care medicine at the University of Illinois, Chicago. And I'm really excited to hear her talk today about pulmonary fibrosis. So please welcome Dr. Kelsey Holbert as today's Grand Round speaker.
Hi, everyone. Good morning. Thank you so much, Andy, for that very kind introduction. I feel very fortunate to be standing here today talking about what I've been working on during my academic time of my chief year and during my research time in residency, a mouse model to further elucidate mechanisms of idiopathic pulmonary fibrosis. And I appreciate you all taking the time to be here today. I have no disclosures. And to start, I'd like to go back to the not too distant past and think about a patient I saw during my intern year. I was on pulmonary consults and met a patient who was newly diagnosed with IPF, who was admitted with a flare. Just before leaving the room, I asked if he had any questions, and it turns out he did. He said over the high flow nasal cannula, "This is not good, is it?" He asked other questions too, like why does it happen? And are there any good treatments? And is there research or any clinical trials? And though I had invited these questions, I did not know their answers. So like a good intern, I told the patient I would talk to my attending and get back to them, and then searched for the answers to his questions as I walked the halls of the hospital, almost certainly lost somewhere on D65. But what I found stopped me in my tracks and confirmed that the patient was right. This is not good. Median survival of just 2 to 5 years. It strikes me that we are here 4 years later, which really does not feel like a very long time ago. In that time, and especially during my chief residency year, I've been fortunate to work in Dr. Sandbo's lab, studying mechanisms of fibrosis. I hope to share what I have learned about idiopathic pulmonary fibrosis in that time, answering some of our patients' questions along the way. To do so, I will talk about IPF broadly, then some of the work that's been going on in the Sandbo lab, and finally my project. By the end, I hope we will be able to meet these objectives, which are to determine the need for additional therapeutic options for IPF, to outline the basic mechanisms of IPF, including extracellular matrix deposition, And finally, to describe a Tensin 1 knockout mouse model for future use in fibrosis research. So first, some background. IPF is a chronic, relentlessly progressive disease which causes scarring and fibrosis of the lungs, which ultimately leads to these changes on chest imaging, and more importantly, cough and progressive dyspnea for patients. These changes cause impaired gas exchange at the level of the alveolus and can eventually lead to respiratory failure. While it is still considered a rare disease, its incidence and prevalence are both increasing. Shown here is a graph from an epidemiologic study of IPF among US veterans from 2010 to 2019, which shows that new cases per 100,000 patients are nearly tripling in this time frame. This next slide I would call the opposite of a busy slide, which shows the just 2 FDA-approved treatment options for IPF, both of which are antifibrotics called pirfenidone and nintedanib. Both of these medications were approved for use in 2014, which is somehow already 10 years ago. In the New England Journal of Medicine study, a phase 3 trial of pirfenidone in patients with IPF, treatment with pirfenidone for 1 year compared to placebo reduced disease progression as reflected by lung function, exercise tolerance, and progression-free survival. In another New England Journal of Medicine study, efficacy and safety of nintedanib in IPF, treatment with 1 year of nintedanib reduced the decline in FVC, which was equated to disease progression. Meta-analyses in more recent years have shown that use of one of these drugs is associated with a decreased risk of acute exacerbations with a risk ratio of 0.63 and a decreased risk of all-cause mortality with a risk ratio of 0.55. However, importantly, these medications do not reverse fibrosis, instead only slow their development. And despite these medications, there remains a high mortality associated with IPF. A 2019 study of the Finnish IPF registry found a median overall survival time of 4.5 years after diagnosis. And you will note that while the Kaplan-Meier curve for the patients who were treated with an antifibrotic, shown in the dotted line, is slightly better than those who were not, there is still about a 50% mortality at 5 years. In addition, though these antifibrotic drugs have been shown to decrease disease progression, their effect on health-related quality of life has not been convincingly demonstrated. So in summary, our patient was unfortunately right. This is a devastating disease, and new options for therapeutics are critical for improving outcomes for our patients. Part of developing new therapeutics for any condition is a good understanding of the mechanisms that cause disease, which we will review now. I will remind us that this is called idiopathic pulmonary fibrosis, so there is still a lot we don't know, but we'll start with what we do. We know that over time, IPF leads to progressive changes of the lung, which have characteristic features on imaging. Here on the right, you see some of these same hallmarks on gross specimens, where we see areas of dense fibrosis in the parentheses and areas with such fibrotic walls that the air spaces have become enlarged, which is called honeycombing, noted by the arrow. And that's compared to interspersed normal lung with no fibrosis or honeycombing and more surface area for gas exchange, shown on the left. So we know that that is the end result, but how do we get here? And to help answer that, we will look at histology. We see a normal lung here on the left with an IPF lung and a circled fibroblastic focus on the right. Fibroblastic foci are areas within the fibrotic lung that contain densely packed fibroblasts, myofibroblasts, and the greatest amount of newly formed collagen, which is key in the development of fibrosis. Fibroblastic foci are important histologically, but also clinically, as numbers of these structures present on surgical lung biopsy have been shown in some studies to correlate with decreased survival. So we now know that the fibroblastic focus is an important component of IPF. But how does the fibroblastic focus develop? And it is thought that recurrent microinjuries to the alveolar epithelial cells, such as cigarette smoke, industrial dusts, GERD, viral infections, and potentially other injuries not yet identified, lead to activation of inflammatory and profibrotic processes in genetically susceptible individuals. This results in increased proliferation of fibroblasts and myofibroblasts, activation of those cells, extracellular matrix deposition, and platelet aggregation in the pulmonary interstitium, which can essentially all be described as an aberrant wound healing process. All of this results in a disruption of the usually tightly regulated structural organization of the lung architecture, and over time leads to impaired gas exchange and organ dysfunction. And because the extracellular matrix deposition is such an important part of the development of fibrosis, we'll look at this in a little more detail. The extracellular matrix, or the ECM, is a complex composite of elastic fibers and collagens that interact and remodel in a usually tightly regulated fashion to maintain the structure of the tissue and allow for normal lung function. In addition to collagen, which you see in the blue fibers here, fibronectin is a glycoprotein that is a key player in directing the organization of the ECM, and additionally in mediating cell attachment and function. This is demonstrated here, where fibronectin is depicted in brown, interacting with a fibroblast, shown in green. But that was all cartoons. So let's see how this translates back to patients. And we can see here we have 3 different IPF lung samples, which were obtained by lung explants or surgical biopsies. And within each of these specimens, there is an outlined fibroblastic focus that is then stained for collagen 1 and fibronectin. And you will note that there is heavy staining of both of these in the fibroblastic focus, with more regular lung architecture and lack of staining in the other areas of the lung. I will also point out, noting that this will have implications when I discuss our work later in the talk, that fibronectin assembly is a critical event in the development of fibrosis. And to help demonstrate that, I will show some older work from a different lab that shows that disruption of fibronectin assembly also disrupts collagen deposition, and by definition, disrupts fibrosis. So this group took fibronectin null cells and plated and incubated them with fibronectin for 16 hours. In this first panel, cells were then fixed and processed for immunofluorescence, and you can see staining for collagen on the left and fibronectin on the right. In this second panel, cells were then washed and incubated with culture medium without fibronectin for 24 hours, and collagen deposition and fibronectin staining were then absent. And in this last panel, cells were similarly washed, but instead incubated with fibronectin-containing media. And again, you see the collagen and fibronectin staining. So in summary, these data indicate that fibronectin is a critical component of this process that regulates the assembly of other proteins into the extracellular matrix. And in summary of this section of the talk, in IPF, aberrant wound healing after alveolar epithelial cell injury leads to excessive deposition of ECM and causes areas of stiffened fibrotic matrices to form, called fibroblastic foci. And these have been shown to correlate with decreased survival. Additionally, fibronectin assembly is a key step in this process. So we have established fibronectin's role in the fibroblastic focus and the fibroblastic focus's role in IPF. And we will now build on this knowledge as we talk about some of the work that the Sandville lab has been doing over the last several years, looking specifically at Tensin 1, which interacts heavily with fibronectin and which we hypothesized to be an important player in the pathogenesis of IPF. I will note that moving forward, when I say we, I mean the Sandville lab, and in many cases really mean they, as a lot of great work was done prior to me joining the lab. However, with their permission and for simplicity, we'll say we. And now back to Tensin 1. Like I said, Tensin 1 interacts heavily with fibronectin, and we hypothesized it to be an important player in the pathogenesis of IPF. But what is Tensin 1? Tensin 1 is a multi-domain protein that has been shown to be involved in many different processes, including cell adhesion, cell migration, cell proliferation, and signaling across the plasma membrane. Shown here is immunofluorescence from our lab highlighting Tensin 1's role in the extracellular matrix. This shows that Tensin 1, shown in green, is a component of two types of adhesions: focal, which are important for making contact between the extracellular matrix and cells, and fibrillar, which are important in assembling the fibronectin matrix itself. And we hypothesize that Tensin 1 may play an important role in nascent extracellular matrix formation in the pathogenesis of pulmonary fibrosis. Importantly, we found that Tensin 1, stained here by immunohistochemistry in brown, is upregulated in the fibroblastic focus of human IPF lung sections, shown on the right, compared to the normal lung on the left, where there is notably less staining. Additionally, lung tissue was isolated and homogenized in a solution that enabled us to evaluate mRNA expression in tissue samples from normal and IPF lung. And this showed that there was increased expression of Tensin 1 in IPF samples compared to normal lung samples. We then looked specifically at fibroblasts, which are the key mediator cells of this process, and found that both mRNA expression and protein levels of Tensin 1 were increased in fibroblasts from IPF samples, again shown on the right of each of these graphs, compared to those from normal lung, shown on the left of each of these graphs. So we found that Tensin 1 is upregulated in the fibroblastic focus of human IPF lung sections and in fibroblasts, the key mediator cells of this process. Further, we found that loss of Tensin 1 resulted in decreased assembly of the fibrillar fibronectin matrix. Briefly, small interfering or siRNA was used to mediate knockdown of Tensin 1 in human lung fibroblasts. When Tensin 1 was knocked down and cells were incubated with TGF-beta to induce a pro-fibrotic fibroblast phenotype, there was decreased assembly of a fibrillar fibronectin matrix, shown in this red box. So saying that again, siRNA was used to mediate knockdown of Tensin 1 in human lung fibroblasts. When Tensin 1 was knocked down and cells were incubated with TGF-beta to induce a pro-fibrotic fibroblast phenotype, there was decreased assembly of a fibrillar fibronectin matrix, where the Tensin 1 knockout group is shown on the far right, and the negative control is in the second from far right. We also looked at a deoxycholate extraction, which is an assay that allows for determination of the amount of extracellular matrix-bound protein. When proteins are bound to the ECM, they become deoxycholate insoluble proteins, which are represented in green in this diagram. And using this assay, we found that the amount of ECM-bound collagen and fibronectin also significantly decreased in the Tensin 1 knockout group compared to the negative control, which is shown by Western blot here on the left and graphically on the right, where the amount of ECM-bound collagen is shown in gray and the amount of ECM-bound fibronectin is shown in black. And there is significant decrease in both of these when Tensin 1 is knocked out, shown on the right. So this work showed that Tensin 1 plays a key role in the assembly of the fibronectin matrix. But this was all in vitro, and we are ultimately interested in understanding how these mechanisms could lead to better patient care. So we were interested in studying Tensin 1 knockout in a more sophisticated model, this time a mouse model, to help further characterize Tensin 1's possible role in fibrosis. With a literature search, a Tensin 1 knockdown mouse model was identified. This was a germline deletion, so mutant alleles were injected into blastocysts and mice were born without Tensin 1. These mice unfortunately developed cystic kidney disease and had abnormal mating potential, which limited their use for our purposes. In addition, these mice were born with a Tensin 1 deletion, which did not allow for studying temporal effects, which can be more translatable to human conditions. So we, again, they, created a Tensin 1 knockout mouse model, which allowed for inducible Tensin 1 deletion. And when I first joined the lab, they told me they created a mouse that used CRISPR-Cas9 technology to flox the Tensin 1 exon 8 and then mated these mice with Cre recombinase strain of mice. And I said, oh no, thank you so much. I am in the wrong place. So if you are anything like me, you might be feeling very confused. So we will go through each of these steps in detail and then come back to this summary slide. The first step is CRISPR-Cas9, which many of you are likely familiar with. You first find your sequence of interest, here Tensin 1. You then design guide RNAs, which are specific to the Tensin 1 sequence, and also have palindromic repeats that allow for recognition of the Cas9 enzyme. This guide RNA guides Cas9, which is a gene editing enzyme, to the right spot to make a cut in the DNA. And you can do different things at this point, most commonly an insertion or a deletion. In our case, we inserted 2 LOX-P sites. LOX-P sites are 34 base pair sequences that will later be recognized by a different enzyme called Cre recombinase, also referred to as Cre. Cre recognizes LOX-P sites and essentially cuts out what is between the 2 LOX-P sites. In our case, we inserted these LOX-P sites on either side of the Tensin 1 exon 8. Tensin 1 exon 8 was chosen because it is conserved across many different tensin variants, which is shown in this figure here, where each of these horizontal lines is a different Tensin 1 variant, and the red exon is exon 8, which is conserved across all of these. This whole process is called floxing Tensin 1. So in order to achieve this, wild-type zygotes were injected with everything that they needed to flox Tensin 1. This included LOX-P oligos, Cas9, and guide RNA. Embryos were then transferred to surrogate mothers, and the pups were then genotyped to ensure that Tensin 1 had been floxed. And then those mice that had the LOX-P sites surrounding Tensin 1 exon 8 were mated with a tamoxifen-inducible Cre recombinase strain of mice. Again, Cre is an enzyme that allows for deletion of a sequence between 2 LOX-P sites. And the tamoxifen-inducible part refers to the fact that the Cre enzyme is only activated when tamoxifen is given to the mice. These mated mice now have both the LOX-P sites and the inducible Cre. Then, when tamoxifen is given to the mice intraperitoneally for 7 days, the Cre is activated and is able to find the 2 LOX-P sites. The activated Cre deletes what is between those 2 LOX-P sites, so Tensin 1 exon 8 is deleted, which causes a frame shift mutation, making the Tensin 1 protein non-functional. And we have created a Tensin 1 knockout mouse. And we did not take these mice to the salon. They really did not change color. But I will use this mouse figure to indicate the Tensin 1 knockout mice going forward. So now that we have gone through each step in detail, let's come back to this summary slide. We use CRISPR-Cas9 technology to flox Tensin 1. The Tensin 1 floxed mice are mated with tamoxifen-inducible Cre recombinase strain mice. And then those mice are given tamoxifen to induce the Cre. And the tensin 1 exon 8, which is between those two LOXP sites, is then deleted, causing a nonsense frame shift mutation, making the tensin protein non-functional. And we have now created an inducible tensin 1 knockout mouse model. After this, we confirmed efficient global knockdown of tensin 1 by mRNA through RT-qPCR, shown on the left, as well as by protein through Western blot, shown on the right. So in summary, we created a mouse model which had efficient, inducible, and global deletion of tensin 1. And that brings us to my project, which was to further characterize this mouse model. And we first wanted to ensure that these mice were healthy. And to do so, we wanted to look at mating potential of the mice, evaluate weight trends as a surrogate for the health of the mice, analyze their blood, and assess the histology of their major organs. We had particular interest in the mating potential and the histology of the major organs compared to the prior model. Before we move on to discuss some of this, I want to explain the nomenclature that you will see here. So WT are non-genetically modified wild-type mice. Tensin 1 flox mice are those that have the LOXP sites on either side of tensin 1 exon 8 using CRISPR. And then Cre-positive tensin 1 flox mice are those that have the tamoxifen-inducible Cre as well as tensin 1 floxed. But I'll point out that all of these mice are tensin expressing at this point because we have not yet given them tamoxifen to induce deletion. So we first looked at the litter tensin 1 flox and Cre-positive tensin 1 flox mice. And we looked at the number of litters per mating pair and the number of pups per litter. And there were no significant differences in either of those measures, so we concluded that these mice had overall normal litters. Like I've said, these mice are genetically modified, but still do express tensin when they are mated, which is a benefit to our model as it allows us to circumvent the mating problems that the prior germline deletion model faced. And now moving forward, the mice nomenclature will change slightly, where mice that express tensin will be referred to as tensin 1 plus or tensin expressing mice, and that the mice that are floxed, have Cre, and were given tamoxifen, and thus have tensin knocked out, are tensin 1 minus or tensin 1 knockouts. We looked at the weight trends between these two groups of mice, the tensin expressing and the tensin knockout mice. All mice were given tamoxifen for 7 days, but only the tensin 1 knockouts had everything that they needed to delete tensin. The percentage change in weight is plotted on the y-axis and days on the x-axis. Tensin 1 expressing mice are shown in the white circles, and tensin knockout mice are shown in the black squares. And again, there was no statistically significant difference between these two groups, this time using two-way ANOVA. There is a slight downtrend in weights after tamoxifen treatment, which is consistent with other studies. We then collected blood from both tensin 1 expressing and tensin 1 knockout mice at 14 days after tamoxifen administration. And there were no significant differences between CBC or CMP between these two groups. Shown here is white blood cell count, hemoglobin, platelets, BUN, creatinine, AST, and ALT. We then anesthetized and euthanized the mice before harvesting the major organs, including lungs, spleens, kidneys, hearts, and livers, and submitted these to a vet pathologist for review. We'll first look at the kidneys, which fortunately did not have any cystic kidney disease. There was some minimal subacute inflammation of the kidneys and serositis, which is shown in the most zoomed in pictures in the bottom right of each of these conditions. And this was felt to be related to some inflammation from the intraperitoneal injections themselves, but was consistent between the two groups. We then looked at the lungs, which demonstrated infiltration of lymphocytes and plasma cells histiocytosis, and perivascular cell infiltrates. But these changes were again found in both the tensin expressing and the tensin knockout animals' lungs. These changes were attributed again to tamoxifen administration, which has been reported in previous studies, specifically a recent paper on the histopathologic changes induced by tamoxifen in Cre recombinase negative mice. Some of their findings included increased alveolitis, vasculitis, and pleuritis, which was similar to what we observed. So we took this to mean there may have been some tamoxifen-related changes, but no tensin 1 knockout-related differences. And there are additionally no significant differences in the hearts, spleens, or livers of these two groups. So from all of these studies, we concluded that the tensin 1 knockout mice were overall healthy animals. After developing the model and concluding that they were overall healthy, we then tried to further characterize these mice and generate hypotheses for future study in fibrosis mouse models. A lot of the work that I have done on this project is around RNA sequencing, so I'll spend some time going through that process as well as explaining the gene enrichment analysis that was able to be performed as a result of the RNA-seq. I then validated some of these findings from the RNA-seq by RT-qPCR, and we'll show that data as well. So RNA sequencing is a newer technology that measures the transcriptions of genes in a biological sample. So we input RNA from the biological sample, and by the end want to estimate the relative abundance of transcripts that come from each gene in the sample. And we wanted to use this method to evaluate the genetic differences that arose when we knocked out tensin 1. We started by isolating RNA here from mice whole lung lysates, and then fragmented these RNA molecules into roughly 150 base pair segments. RNA-seq utilizes reverse transcription to transcribe RNA to cDNA, which is amplified before being processed with a sequencing machine. We worked with the University of Wisconsin Gene Expression Center to then sequence all of these fragments. And we did this using an Illumina NovaSeq system, which allowed for an output of 66 million reads per sample. These reads are then aligned to the known mouse genome and then quantified. In the quantification step, the control and experimental group are compared to each other. For each gene, the number of reads that align to the gene are counted, and a relative abundance or a differential gene expression is reported between the two conditions and presented in table format, very big table format. In our study, over 15,000 genes were reported and over 1,600 genes were found to be differentially expressed with a p-value less than 0.05. However, due to this large sample size, an adjustment called the Benjamini-Hochberg adjustment was applied to reduce the chances of false positives. Of these 1,600, 171 were then found to be differentially expressed with an adjusted p-value or false discovery rate of less than 0.05, which is a particularly stringent method of analysis. Compared to the tensin expressing mice, 68 genes were found to be up and 103 genes were found to be down in the tensin 1 knockout condition compared to the tensin expressing mice. All of these were then entered into the string database, which is an online database of known and predicted protein-protein interactions that allows for functional enrichment analysis. And functional enrichment analysis is a method of identifying classes of genes that are overrepresented in a large set of genes and creates a network of interconnected genes, which can help you process and analyze larger data sets like ours more meaningfully and generate hypotheses. So we entered the 171 genes and 154 were recognized by the database, and we got a network that looked like this, which is a little bit overwhelming. From here, we then use cluster analysis to help organize genes into clusters based on how closely associated those genes are. With assistance from our biostatistician, we applied Markov clustering, And we were left with 11 clusters, which you can see represented by the different colors here. We then looked more closely at each of the clusters, and I will discuss the top 2 networks, which we found some interesting and surprising findings. But first, some orientation to this view of the clusters. So each of these rings represents a differentially expressed gene. The blue rings are those that were downregulated in the tensin 1 knockouts compared to the tensin expressing mice. The red rings are those that were upregulated in the tensin 1 knockouts compared to the tensin expressing mice. The size and color of the ring correlates with the differential expression. So the larger and the darker the ring, the more up or down that gene was in the tensin knockout in comparison to the tensin expressing mice. The thickness of the line connecting the nodes is a string feature that indicates the degree of confidence in the prediction of the interaction. And I know I kept you all on the edge of your seats with the surprising findings, so now on to those. We are again looking at the first cluster from our MCL clustering, and the first interesting finding was that we saw a downregulated myeloid signature in the tensin knockout mice. As a reminder, myeloid lineage cells include platelets, monocytes, including macrophages, and neutrophils, among others. We can see that several important myeloid markers were downregulated in the tensin 1 knockouts compared to tensin expressing mice. Some of these include the matrix metalloproteinase MMP12, chemokines like CXCL2 and CXCL9, and other key myeloid markers, including TREM1 and MARCO. 2 of these were validated by RT-qPCR in the same and a separate cohort of mice and were statistically significantly downregulated in tensin knockouts as well. Another interesting finding was that there was an upregulated B cell signature. Some of those genes included CD19, CD22, SPI-B, and BTLA. And again, some of these were validated by RT-qPCR in the same and a separate cohort of mice and were statistically significantly upregulated in tensin 1 knockouts as well. So in summary, RNA sequencing was used for hypothesis generation about this newly created mouse model. And while we had previously studied tensin 1's role in the focal and fibrillar adhesions as well as in fibronectin assembly, we were surprised to find that in addition, tensin 1 may have a role in modifying the immune system response. And further, we hypothesized that tensin 1 could be a critical component of homeostatic signaling in immune and myeloid cells. But like I said previously, we want to be able to use this mouse model to better understand the mechanisms of idiopathic pulmonary fibrosis. So in the last section of the talk here, I will talk about 2 of our ongoing studies that further evaluate the tensin 1 knockout mouse model in the context of fibrosis. We use a bleomycin-induced fibrosis model that has been well studied in the context of pulmonary fibrosis. In this model, we give the mice 1 dose of intratracheal bleomycin, which results in the alveolar epithelial cell injury, which I discussed previously. Following that, there is an inflammation phase for about 5 days, after which we see the fibrotic phase start to develop, which is characterized by myofibroblast formation, with around day 28 being peak fibrosis. This is a self-resolving fibrosis model, so after that point, there begins spontaneous resolution. And for our model, in order to induce deletion of tensin 1, we give tamoxifen for 7 days prior to the bleomycin installation. For this study, we used these 4 conditions. We had a tensin expressing mouse, shown in green, and a tensin knockout mouse, shown in red, that received intratracheal bleomycin to induce fibrosis. We also had a tensin expressing mouse, shown in blue, and a tensin knockout mouse, shown in yellow, that received normal saline intratracheally as a negative control. And with this experiment, we wanted to find out, is tensin 1 knockout protective against the development of fibrosis in this model? And what we found was that yes, tensin 1 knockout was protective against fibrosis. We evaluated this using the hydroxyproline assay, which is a way to quantify collagen deposition. We again used whole lung homogenates for this assay, and we saw that there was significantly decreased hydroxyproline in the tensin knockout mice, again shown in red, compared to the tensin expressing mice, again shown in green, that both received bleomycin to induce fibrosis. Looking at this with a different model, shown here are 3 lung tissue sections that were stained with Mason's trichrome technique, which stains for collagen deposition. We did not include a tensin 1 knockout saline group in that study. So in the blue panel is the normal saline treated tensin expressing mouse, which served as our negative control. In the green is the tensin expressing mouse that received bleomycin to induce fibrosis. And the red is the tensin 1 knockout mouse that also received bleomycin to induce fibrosis. But you can see that when the tensin expressing mouse was given bleomycin, again shown in green, there's quite a bit of dark trichrome staining, which again stains for collagen deposition. But we do not see that same level of staining in the tensin knockout mice that also received bleomycin, shown in red. So overall, tensin 1 knockout was protective against fibrosis in this model. But this is when we knock out tensin 1 before the mice received bleomycin. And we were also interested in seeing whether knockout of tensin 1 after the mice received bleomycin was also protective. Since you can imagine, this may be more translatable to human conditions, since people come to us already with fibrosis. So we use the same model, but slightly modified, where the tamoxifen was given to knock out tensin on days 5 through 11, after the mice had already received bleomycin. And we saw similar trends where when evaluated by hydroxyproline assay, there's a decreased amount of collagen deposition in the bleomycin-treated tensin 1 knockouts compared to bleomycin-treated tensin-expressing mice. So in summary, tensin 1 knockout was protective against fibrosis if tensin 1 was knocked out before bleomycin was given and after bleomycin was given. So this work lent support to our previous hypothesis that tensin 1 was a key player in fibronectin and collagen deposition and ultimately fibrosis. But through the RNA sequencing, we had learned that tensin 1 may have this role in immune system response. So we next sought out to further characterize this. And for this, I did a Luminex assay, which is a magnetic bead-based multiplex assay that allows for simultaneous analysis of multiple chemokines and cytokines with just microliters of fluid. Sample is loaded in a 96-well plate, and each sample contains magnetic beads that are specific to different cytokines. You then add a series of reagents to allow for fluorophore detection of each cytokine. We again used the same 4 mice conditions and collected bronchoalveolar lavage fluid 3 days after bleomycin instillation. We looked at the secretion of 32 different chemokines and cytokines in these samples with this multiplex assay. And we found that in bleomycin-treated tensin-expressing mice, again shown in green, compared to normal saline-treated tensin-expressing mice, shown in blue, there was a statistically significant increase in many cytokines, including colony-stimulating factors, interleukins, and chemokines. There was a decrease in IL-2. And we were not particularly surprised to find these mostly increased levels of cytokines in the inflammatory phase of the bleomycin model. However, on our literature search, this has not been previously well characterized. What we were also interested in finding out, however, was if there was differential secretion between the bleomycin-treated tensin 1 knockout mice and the bleomycin-treated tensin-expressing mice, again, the red and green comparisons. Said differently, we were interested in knowing if upon bleomycin treatment, tensin 1 knockout affected cytokine secretion. And we saw that of the 13 that were up in the bleomycin-treated knockout mice, 4 were further upregulated in tensin 1 knockouts, including GM-CSF, CXCL10, TNF-alpha, and IL-4. The last of these 2 were not statistically significant in this limited number of samples, but trending, with a p-value of 0.06. Showing this graphically compared to the negative control in blue, which was tensin expressing normal saline treated mice, you can see upregulation in the green, tensin expressing bleomycin treated mice. You can also see further upregulation in the tensin knockout bleomycin treated condition, red, compared to the tensin expressing bleomycin treated, green, for each of these 4 markers, GM-CSF, CXCL10, TNF-alpha, and IL-4. So again, while we had previously studied tensin 1's role in fibronectin assembly and the focal and fibrillar adhesions that I showed previously, we are surprised to find that in addition, tensin 1 may have a role in modifying the immune system response in the context of a fibrosis model this time. So as we wrap up here, I would like to summarize again what we have found through this work. One of the main hallmarks and known mechanisms of IPF is excessive extracellular matrix deposition. Because we knew that fibronectin assembly was a key step in this process, we developed an interest in another player that is also involved in fibronectin assembly, tensin 1. We then developed an elegant mouse model that allowed us to study this further. And from here, we were excited to find that the tensin 1 knockout was protective against fibrosis. Through RNA sequencing, we were surprised to find that tensin 1 may also be modifying the immune system response. And because of these findings, we have started to use Luminex and other methods to further characterize how tensin 1 may be modulating the immune system response in fibrosis. Overall, we believe this work leads to a more nuanced understanding of the key mechanisms that contribute to pulmonary fibrosis. So, as we look back to our patient from my intern year, I think we have addressed some of our patients' initial questions. And here I am still with many questions, but different and exciting questions. And I think part of why I like research so much and why I'm excited to continue doing research in my career are these unexpected and surprising findings, which allow us as clinicians to have a more complete understanding of the diseases we treat, and that'll allow us to look for future answers to our patients' questions. As I was preparing for this grand rounds, I was able to reflect on how much I have learned in these 4 short, sometimes very long years. And that is in large part to so many people, some of whom are listed and pictured and here in the audience today. I want to say a special thanks to the Sanbo Lab and all of the work that they have put into this project, and for teaching me so much in the last few years, and for bearing with me being a resident and also working in the lab. And lastly, for creating an environment that allowed me the confidence to pursue being a clinician researcher in the future. Special shout outs to my wonderful husband, Derek, and our daughter, Nora, who has brought us so much joy this year, and the rest of my family who have supported me endlessly. Thank you to my co-chiefs, Amy, Carolyn, and Mike, whom I have tremendous respect and admiration for. Thank you to these listed and other mentors who have supported my career so far, particularly the outstanding leadership from Dr. Coyle, Dr. Sambo, who is working clinically today, but here virtually, Dr. Bernau, and Dr. Schnapp. Here I have some additional acknowledgments, particularly our admin staff and Catherine, many of whom are also here today. And lastly, gratitude for the funding that has supported this project. These are my references, and I am happy to take any questions.
All right, thank you, Kelsey, for a wonderful grand rounds. Open to questions from online or in the audience. All right, Rob, making me walk far on the first one.
Well, microphone's coming up kind of loud. I just want to really, it was a great talk, but I was particularly impressed that you showed some vulnerability that a lot of this stuff is kind of scary and you need to be willing to be dumb, which a lot of physicians aren't always willing to do in order, but you know, clearly you made a lot of progress. All right, so my question is, you mentioned that that there are variants in Tensin 1. Have any of these variants been associated with altered susceptibility or increased prevalence in IPF?
Yeah, thanks for the question. I have certainly gotten good at admitting what I do not know, which is a lot. But yes, so there was a genome-wide association study not too long ago that looked at a bunch of different SNPs, but they actually found a Tensin 1 SNP that was associated with decreased lung function across many different conditions, including IPF as well as COPD. So there's some ongoing work in our lab actually looking at some of those SNPs in IPF samples specifically.
Other questions from the audience? I see there's one online as well.
Hey, Kelsey, great job. And I'm going to take a totally different question approach here. But so now with your unique perspective of this tremendous success with Dr. Sandbo's lab and some of the collaboration, but your role as a chief in the education, when you look back to your intern year, early soft, second year, would you do, or how would you want a program to start to introduce these concepts to trainees? Like when you look, how do we best do that to get people such radically different worlds? How do we start to pair them and get people engaged, interested? Just want to see your take as a retiring chief.
Yeah, I love that question. I think that is a great question to think about in your chief year as you have kind of an oversight of some of what goes on in the curriculum especially, and can have some perspective on how busy you really are as a resident and where this all might fit in. I think that there's so much outstanding work that is going on at UW, and I think sometimes the residents are like siloed away from it because they're busy working in the middle of the night and such. So I think making connections has been the way that I have gotten involved with some of this work. So I think we have such awesome work going on and so many amazing mentors and some awesome residents as well. So being able to pair those together in some way early in training might allow for some stimulation and some collaboration. I think like the first question had said, I think it's kind of scary to get involved in research, or many of us feel like we don't have all the skills that you might need to contribute to a project like this. I have certainly felt that many times, but I think that we all have unique skills that can contribute in a research or a lab or any sort of project that come through our training. And I think in our lab, we've been able to collaborate in really unique ways, like things that I can bring from the clinical side and things that everyone else can teach me other things from the other side. So I think early exposure, connections, and then maybe just having a little bit of faith that you do have the skills that you can bring to a lab, and you'll learn much more than you can bring, but that's okay too.
Thanks, Kelsey. That was really awesome. So I guess two questions. The biologic question is, and maybe I missed this, is Tensin 1 expressed in the bone marrow, and could that explain the myeloid results that you saw during the early characterization phase? And then I was so impressed by how well you explained the methods. It was really, I learned stuff too. I mean, but have you thought and reflected about how you communicate even basic science to patients?
Yeah, that's a great question. Something I think about a lot. I think actually that this part and like communicating our findings and making figures and posters and things like that is actually, I think, one of the most fun parts. And I think like there are people who are trained in showing it in a more easily understandable way, like design people, et cetera. So I think I still have a lot to learn in that aspect. But thinking about how we can present this to patients, yeah, it's a great question. I think I think it requires that you have a very good understanding of it yourself. And I think preparing this, like I also learned things about the things that I have been working on. So I think in any work that you want to pass along to a patient, the more you understand it, the easier it is to kind of pare it down. So I guess that is one thing that I have learned with my research. I'm not very well able to communicate it if I don't really understand each piece of it. So trying to have, like taking that time to really understand it allows you to kind of think about it in a more broad picture. And then, you know, there may be other people outside of medicine, outside of research or basic science research that may be able to help us kind of communicate things better that we can collaborate with in the future. And then to answer your first question about whether Tensin 1 is expressed in the bone marrow, I do not know the answer to that. Ksenia, Dr. Brunau has been looking for that answer, I think. But we'll find out and we will get back to you. But that's a great question.
Great. So we have one question from the virtual audience. And then before that, one shout out from Dr. Sam, who said, awesome work, Kelsey. So nice shout out in the chat. This one's from Dr. Lamming. Said, great talk. Said the effects of Tensin 1 deletion on fibrosis in the bleomycin model are very interesting. It is similar to the effects of mTOR inhibition. Do you know if mTOR signaling is suppressed by Tensin 1 deletion?
Have not looked. Something we will consider for the future. Thank you for the question, though. I appreciate that.
Always new things. One question about sort of IPF itself. Are nutritional supplements being looked at as playing a potential role to combat inflammation to prevent progression or development of pulmonary fibrosis?
Have any pulmonary clinicians, Dr. McCartney?
One of the areas that's always so interesting was earlier therapies, right? Therapies that really not studied, really toxic. And so there was a big push of antioxidant nutritional supplements, particularly in acetylcysteine, that really was going to be the key. The problem was that what the real studies ultimately showed was that acetylcysteine actually was just kind of minimizing the effects of toxic therapies. People did better in the study because they're killing those cyclops. So it's a loaded subject, and you're still going to see people that are taking acetylcysteine. There's not great data that by itself. You're not on these other therapies. There are believers out there.
Thank you, Dr. McCartney.
Maybe time for one more question if we have it. No, watch maybe two. We'll do two and then we'll finish.
Hi, Kelsey. Thank you so much for this talk. I just had a brief question, and I know it takes a long time to really come up with an experiment, but do you guys ever looked at kind of first inducing, I guess, fibrosis with bleomycin and waiting kind of a longer time to have more established fibrosis and then inducing like, you know, your knockout with tamoxifen and see if there's any reversal of that fibrosis?
Thank you for the question, Oscar. And I'm realizing as I'm looking at my slide that I put on there to thank the residents, but I did not thank the residents. So I'll take this moment to thank the residents. Working with the residents has been by far like the best part of Chief here. So thank you for the question. Thank you for being here. And thank you to all of the residents. Sorry, I failed to mention you. Getting back to your question about whether we had looked at different time points for in deleting Tensin 1 in the bleomycin fibrosis model, we have looked now at 14 and 21 days. And yeah, it's one of the problems with the bleomycin model is that there begins that spontaneous resolution at 28 days. So there's sort of a limited time point or like a limited time frame where you have fibrosis and can actually study the fibrosis. So the models that we've done so far are sort of the latest where we see that peak amount of fibrosis before resolution starts to develop.
We are right at 9. So I want to thank Kelsey for a really terrific grand rounds. You did a wonderful job explaining really complex scientific concepts in a way I think we could all sort of grasp and take lessons away from. So thank you, Kelsey. Let's give her one last round of applause for our terrific grand round.